so many distinguished lecturers. It's really flattering. And I would like to tell you about a lot of the stuff that I will tell you is about our current research in macrophages, studying cytokine expression, IL-10, and TNF-alpha, and the role of cyclic cAMP there. But, however, since this conference is dedicated to the memory of Tzvi Zellinger, sorry. I would like to give a bit of a different type of lecture, not describing only our current research, but starting with the work that I did, or some of the work that I did in the lab of Tzvi Zellinger. That would be the first two topics, and then I will continue to show you some other projects also in my postdoc period, and then some of the projects that we're working on in my lab. So the title of the talk would tell you that most of it is about cyclic cAMP. It's not all of it. Most of it is directly related to cyclic cAMP. Some just stemmed out of the cyclic cAMP research or indirectly related to it. So I will first start with the first project, actually, that Tzvi gave me when I came to his lab as an undergraduate student. And he said that there is a huge complexity in the GPCR's field. Sorry, my throat is not functioning well today. So if you look at the red arrows, you would see that many receptors can activate more than one G protein. And it was a big debate in many fields which G protein is activated by each stimulus or each receptor. So the task that Tzvi gave me was to develop a tool that would enable us and everybody else studying GPCRs to indicate which G protein is activated in each cell type or in each stimulus. And the tool that we developed is a photoaffinity label, which is composed like that. It's the GTP is in purple, and then you have this nitrogen linker. There is radioactivity on the gamma phosphate, and we have this green moiety, which is a photolabile group. So when we irradiate, the carbonyl here opens up to a B radical, and then it inserts itself into any carbon which would be next to it, meaning that when the GTP is in the binding site of the protein, it will make a covalent bond, and we can isolate the protein, and detect which protein was activated. So this is the name that we call this molecule, and I will just show you the best example that we have, that we published, and it is in the visual system of the fly. So in the visual system, well, as you know, in vertebrates, it's transducin. It was already very well known. But in the fly system, the signaling is different, and it's not transducin. It was suspected that it is a GQ protein, which is downstream to rhodopsin. And the way that flies see is also different. They don't have the spectrum that we have. The red light, they don't see at all, so it's like darkness. And the blue light is what we illuminate them in order to activate the cells. So blue light is light. Red light is darkness. It's a bit confusing, sorry. So as you see here, this is just an autoradiograph of the membranes of the fly eyes that we incubated with our probe, GTP analog, and we stimulated them with the red light in lane number 1 or blue light in lane number 2, and then just activated the probe by UV light, and we could run this on a gel and see that there was a dramatic increase in the labeling of this about 40 kilodalton protein, which was about the same molecular weight as we expected from the GQ protein. We also did a blot, but that was still not the conclusive answer, because we have to identify that indeed the specific GQ that is present in the fly eye cells is the one that is labeled. And for that, we raised an antibody. I went with Tzvi to the facility. I remember it very well until now. I went with him to the animal facility here at the Hebrew University, and he taught me how to work with the rabbits, and we made this antibody that was specific for the GQ, which is not like our GQ. And we just did the same protocol of stimulating with either red light, which is darkness, or the blue light, and then immunoprecipitated the GQ protein in the fly. And as you can see here, we indeed found that there is labeling of this specific GQ. Now, if we compete the probe with GPP and HP in number 3, both of these images, you see that we completely compete out the labeling. If we use APP and HP, there is no effect. So we were very happy with this. It was, well, today people are not doing it. We thought that many more people would do it. I think that perhaps people are biologists, are kind of reluctant of using, synthesizing their own molecules. They like to buy it usually. In this case, we were very happy with it because we designed a very simple chemical reaction. It was, we called it one-pot reaction. We just mixed the reagents, we got nearly 100% yield, and we didn't have to get rid of all the other side products or whatever we had there. We just put it on the cells and it worked beautifully. But, however, one reason that it wasn't used as much as we hoped was the chemical synthesis that frightened people. The other reason maybe is because it wasn't working so well with other cellular systems where the difference between where the basal activity is higher. Okay, so in this case, it was beautiful. When we tried to label the GS protein in other cell types, it was not so impressive. It was okay, but not impressive. So I would like to show you now, since this is regarded as school, I would like to show you how this thing can be done now. It's actually a very recent paper just from this month, I think. It was published by the group of Lambert. And they use a more sophisticated, much more sophisticated method in order to answer exactly the same question. Which G protein is activated when I give the cell a specific stimulus? So they use molecular biology. The heterothermeric G protein, as you know, is alpha, beta, gamma. The alpha has the GTPase domain and then the helical domain, HD. And they designed a mini G protein, that's how they called it, mini G protein probe, which doesn't have beta gamma. It only has part of the alpha, the GTPase domain, and the GTP binding is actually uncoupled from the receptor activation. So this probe stays on the receptor. It doesn't detach from the receptor. So you can really monitor it. It's for an extended period. And it also has a fluorescent tag, of course, and nuclear export signal. Most importantly, this yellow part is the fluorescent tag. So you can see here, for example, the beta-adrenergic receptor, beta-2 adrenergic receptor, which is localized only on the membrane. Very beautiful. Of course, it doesn't depend whether it's resting cells or whether it's isoproternal stimulated cells, the agonist. While the mini G protein, mini GS protein that was used here, you can see that it's cytosolic in resting cells. However, in the stimulated cells, isoproternal stimulated cells, it looks identical. The localization is identical to that of the beta-adrenergic receptor. So, and here is the quantitation. It goes to the membrane, from the cytosol to the membrane. And they did the same with all other groups, types of G proteins like GI and GQ and G12 that you can see here. And if you look at the concentration, we usually, when you use isoproternal, we use the concentration of between 0.1 and 1 micromolar. You can see that at 0.1 micromolar, the blue curve, that's the GS activation. It's almost full, while the GI is very, very low. However, if you go to 10 micromolar, for example, you want to be sure that you activate, you put much more isoproternal than you actually need, and then you get another pathway being activated. GI is activated almost to the same extent as GS, and even you can see about a third of the maximal activation of the GQ. And if you look at different ligands of this receptor, they really were able to recapitulate the versus ligands, what they do to the receptor. So the agonist, the full agonist, are the black curve and the gray curve, and then they have the partial agonist, the weak agonist here, and even they see very well the inverse agonist, lowering the activation of these mini G proteins. So until now I talked about the on reaction, and now I'll move to the second project that I did in Tzvi's lab, and that deals with the off reaction of the GTPase cycle. I will not talk much about this slide because I will leave Danny to talk about it. He's the one that actually did this work, but just to be on the same line, everybody would be in the same line. I would remind everybody that the hormone, for example, adrenaline in this picture, working on beta-adrenergic receptors, activate the exchange, the on reaction of the G proteins, meaning GDP is going out, GTP is going in. And then we have the activated G protein, GS protein. In this case, the alpha subunit can activate adenylate cyclase, so cyclic cAMP is produced. And then the off reaction is the GTPase reaction, where the phosphate is cleaved, it goes out of the protein, and we still get the GDP stays inside. And this is the inactive conformation. It can no longer activate adenylate cyclase unless there is a hormone still there on the receptor, and then it could go another cycle and another cycle. However, if you block the GTPase reaction, for example, as Danny and Tzvi showed with cholera toxin, you keep the G protein in the on activation with GTP bound all the time. And of course, if you have a mutation in one of the main two catalytic residues in the G protein, you get the same story exactly. Persistent activation, cyclic cAMP accumulates, and it just doesn't depend anymore on the hormone, of course. In the case of the pituitary and thyroid glands, since the GS protein participates in the growth signal, you get also a very large percentage of tumors that show exactly this type of mutation in either glutamine or the arginine, which are the catalytic residues in the heterotomeric G protein. So the GTPase reaction is, well, the chemistry is relatively simple. If you just look at the chemical reaction now, and it will be more complicated when I show you the protein, of course. So the water molecule, in order to be a nucleophile, has to give a proton to a base, and then the hydroxyl group would attack the phosphate. It would form this bipyramid, which is a transition state, and then the bond with the GDP, with the oxygen, bridging oxygen, would break, and we will get GDP and the phosphate that would go out. If we want to mimic the transition state, we can use aluminum fluoride in combination with GDP. So the aluminum sits exactly in the place of the gamma phosphate, and it's considered a very good transition state analog. Okay, so how did we measure the cyclic cAMP response? We did it exactly as Danny did, about 15 years difference between us in Tzvi's lab. We looked, the reason that we did this project was to develop, the first reason was to develop improved photoaffinity labels. And every time that I finished synthesizing a probe, I would go to the membranes, extract membranes, and do a cyclase, adenylate cyclase assay in order to see that the probe can bind the G protein, it is active, and we actually did two types of assays. So the membrane includes 3 proteins that are relevant for us: the receptor, the G protein, and the adenylate cyclase. And in order to get cyclic cAMP produced, we need 3 small molecules. One is the agonist, which we use the isoproterenol or adrenaline. The other one is the GTP, or the GTP analog that we're testing, its activity. And the third, of course, is ATP, which is the substrate for production of cyclic cAMP by the enzyme. The 2 types of assays are called activation and persistent activation. So activation is a very simple assay. We just put the membranes with these 3 small molecules, and we get cyclic cAMP produced, and we measured it by including alpha-labeled ATP as a substrate to produce that cyclic cAMP, radioactive cyclic cAMP. In the second assay that we call persistent activation, we divided it into 2 steps. So the first step, we put on the membranes the agonist, so the receptor is turned on. We put the GTP analog, so the G protein is loaded, but we don't put the ATP, so there is no cyclic cAMP production. So the system is like waiting that we put the substrate. And then in the second step, we put the ATP, but at the same time, we also put the antagonist. So it can only do 1 round. The GTPase cycle can only do a single round if, the GTP or the GTP analog is hydrolyzed, because it cannot, if I go back again, it cannot do another cycle. Okay, there is no hormone, there is an antagonist here. So we can measure whether there is persistent activation or not. And you can see indeed that if we use GTP, there is very good activation in red. There is no persistent activation because the hydrolysis takes a few seconds, while in this assay, step 2 is something like 10 minutes. Okay, so there is a very, very short period that there is cyclic cAMP production, and then it stops. Now we tested these 2 analogs that I prepared, and as I said, the reason that I prepared these analogs was that I wanted to improve the photochemical yield, and we thought about the chemistry here, and we actually were right about this analog on the right. It is a better photo affinity label. We're actually wrong about the left one. This doesn't have better photochemical properties, but since I already synthesized it, I put it also in the assay, and this was the surprise, that we expected to see this type of behavior that we saw with what we call monoamino-benzophenone GTP. So this group is the benzophenone, and the amino group links it to the GTP, MABP GTP. And it is persistently activating the system, which is exactly what we expected, because every GTP analog that existed until we made this analog was always, when the modification was on the gamma phosphate, it was always hydrolysis resistant. For some reason that at that time we didn't really know, but later we had some insight into. And I will show you that. So the surprise was with this analog, diamino-benzophenone GTP, DAB-GTP. So there is an extra amino group here, and it behaved exactly like GTP. Okay, it was being hydrolyzed because there is no persistent activation, only activation in the regular assay. So I was going actually to trash this molecule because it wasn't a better photoaffinity label, but we had a brilliant idea, and this idea was, we had a very good timing for this experiment, because at that time, just a few months before, Paul Ziegler published his paper about the structure of a G-alpha subunit in the with the transition state analog, with GDP and aluminum fluoride. And he talked a lot about the catalytic residues that are involved, in particular the role of the catalytic glutamine, which from the structure, of course, you cannot know what it is. You can just say that it certainly has a role. Of course, there are all the mutants. We know that it must have a role. But what is this role? So Tzvi thought that since we have on the substrate itself this carbonyl and this amine group, together they resemble the glutamine. So I know your faces are like my face when he told me that, so what? Okay, so it looks like the glutamine, but first we have the glutamine in the protein, so what's the problem? But we said, well, maybe the glutamine is not working, and because of this modification that we put here, it's displaced. And then with all modifications that we do, we don't have activity because the glutamine is not functioning, but because we have here on the substrate itself the amino group and the carbonyl group, it replaces, it substitutes for the role of the catalytic glutamine. So I went on to test it, and indeed Tzvi was right. So this is the structure of Paul Ziegler showing the glutamine, which is in a perfect place in order to coordinate the attacking water molecule, in order to stabilize the transition state. So I remind you, this is like the gamma phosphate, the aluminum, and this is like the oxygens, the fluorides are like the oxygens of the gamma phosphate. And clearly the glutamine is very important here with its interactions. So we term this, the concept that we made in order to describe what we think is happening is substrate-assisted catalysis. And it goes like this. So in the regular protein, the wild-type protein, you have two catalytic amino acids, the arginine and the glutamine. If you mutate, for example, the glutamine, in this case, you get an inactive protein. And if you just introduce a different substrate, not the GTP, but the GTP with the appropriate chemistry that can substitute for the missing residue, you can gain the activity again, but only on this substrate. So this would be substrate-assisted catalysis. And I would skip all the data just to show you the final result. The blue numbers are the real values, but for simplicity, let's look at the red numbers, which is normalized to the intrinsic GTPase reaction, chemical rate of the GTPase reaction in water. So the GS alpha protein stimulates this reaction 50,000-fold. And you can see that when we test our probe, the DAP-GTP, we have about the same rate. Now, if we take a mutant G protein where there is no glutamine, no catalytic glutamine, we lose completely the activity of the protein on GTP. So I just gave up after about 70 minutes because there was no hydrolysis at all, nothing at all. If I would continue for 10 hours, I believe that I would write here less than 9 rather than less than 90. So really inactive completely. While for DAP-GTP, this was striking. It was exactly the same rate as we got in the wild-type protein. So indeed, it is substrate-assisted catalysis. Only on this substrate, we got the activity. So we were able to rescue this protein. I remember that once I got it, a few days later maybe, I talked with Roger, who was every summer working. I was lucky to be working in the same room with Roger every summer when he came to Jerusalem. So we hosted him, gave him space, lab space, and I showed him these results, and Roger immediately asked me, so what is the mechanistic conclusion? And the mechanistic conclusion of this is that if we have exactly the same rate for a different substrate, not for the same substrate, and we say that we are substituting these groups for the glutamine, so it necessarily means that the glutamine does not participate in the rate-limiting step of the reaction. Otherwise, it must be a different rate. And then we worked with Boaz Sha'anan, who did the modeling on this structure of Paul Ziegler. So you see here the overlay, actually, of the crystal structure of transducin with GDP and aluminum fluoride, the transition state analog in green, and then in cyan, it's the molecular modeling that we have done when we just added on top of the GTP that you see here, the gamma phosphate or the aluminum here, and then we added this part, which is the diaminobenzophenone. And then we let it work in the computer and see what is the ensemble of minimal energy conformations. And you can see that it matches very, very well. It overlaps completely except for two residues. This one is not interesting because the side chain is pointing away from the active site to the solvent. But this is the catalytic glutamine. And of course, it was no surprise for us. This is a zoom. So you can see again the alpha, the phosphate, alpha phosphate, the beta phosphate, the bridging oxygen. This is the aluminum, like the gamma phosphate. You see the distances with the catalytic arginine and the threonine, and this is the attacking water molecule. This is the magnesium that we need here. And the catalytic glutamine sits in hydrogen bond distances to where it is supposed to be, coordinating the water molecule and the oxygen of the gamma phosphate. So 2.5, 2.6, sorry. Okay. You can see in the model, in the molecular modeling, we got preserved all the distances with the arginine and the water molecule, the magnesium, the threonine, everything is the same, except for the catalytic glutamine, which is now 2 angstrom more, which is completely out of the range of the hydrogen bonds that are needed there. So it supported very well our studies, our functional study, that indeed the catalytic glutamine is displaced whenever we have a GTP analog, which has a modification, bulky modification on the gamma phosphate. However, if we have the right chemistry here on the substrate itself, we can get activity again. So the amino group sits exactly where we want it to be. The carbonyl group is pointing away, so this suggested to us that it's not participating in the reaction, and that was our next project indeed, where I synthesized with the help of 2 undergraduate students and a master's student, actually, one of them became, 9 more analogs. So basically it's the GTP and this nitrogen linker and the aromatic ring, and in positions 1, 2, and 3, we just substituted for different atoms or groups, and the purpose was we had a few possible mechanisms. So each of these analogs was supposed either to support or to refute one of the mechanisms. So we covered all the physical, organical, chemistry knowledge that I gained when I was a chemistry undergraduate student, and we made all these analogs. We got help also from Michael Chorev, who advised us about it. And I will cut the story short again. The final conclusion that we had out of all these analogs, that we indeed need only the amino group, and the function of this amino group is to donate a hydrogen bond. So we suggest that the amino group of the glutamine as well is participating in the reaction by polarizing the water molecule to attack the gamma phosphate, and then stabilizes the transition state which is formed. So I will quickly go to the last project that I did in Tzvi's lab. Thanks to Dudi, I don't have to explain you about RAS now. So I will just say again that RAS is a small G protein. It has the same GTPase cycle in essence. And Dudi talked a lot about how common these mutations are found in tumors, something like an average of 30%, but it can go to 90%, as you showed in pancreatic cancer, for example. And what I want to add to what Dudi said is that all of these mutations, or almost all of these mutations, are in either the catalytic glutamine or the G12 mutation that you also mentioned. So we asked ourselves whether we can do the same substrate-assisted catalysis with the RAS protein using this probe that we had, this special GTP analog. And for that, I contacted Fred Wittinghofer in Germany, in Dortmund, who I regarded as Professor RAS. He had a lot of accomplishments about RAS mechanism and small G proteins in general, not only RAS. And I worked specifically with Reza Ahmadian in his lab, who was a postdoc at the time. And the results that we got were really striking, because in this G protein, the rate of GTP hydrolysis and DAP-GTP hydrolysis was different. DAP-GTP was undergoing hydrolysis much faster, 10-fold faster than GTP. Okay, the green line versus the yellow dots. Then we went, this was with wild-type RAS. And then when we went to the glutamine mutant of RAS, the oncogenic mutation, the difference was even much larger, because GTP was, of course, the rate of GTP hydrolysis was reduced by an order of magnitude to almost 0. I mean, this is really, really low. It's almost nothing. While DAP-GTP was hydrolyzed exactly the same rate as in the wild-type enzyme. Okay? No difference at all. So 2 orders of magnitude between GTP and DAP-GTP. And then the biggest surprise was when we looked at the G12V mutation, the most commonly found oncogenic mutation of RAS. And over there, of course, GTP is hydrolyzed very, very slowly. DAP-GTP, not only that it was still hydrolyzed, it was even hydrolyzed much faster, about an order of magnitude faster than in the wild-type protein. Somehow the conformation of the active site allowed the substrate substrate-assisted catalysis to be perfect, to be optimal. So we did many more biochemical experiments to support it that I will not show you, with another system, another type of assays. This is also a crystal structure that was done over there in Germany that shows very nicely that the modification that we have on the substrate itself, okay, so GTP, this is the guanine, alpha phosphate, beta phosphate, gamma phosphate, and this is the benzophenone group, and you see how it displaces the glutamine. Doesn't let it act, function in this reaction, but the amino group is exactly where it should be in order to substitute for the function of the catalytic glutamine. So I didn't show you all results. We showed that it's hydrolyzed exactly at the point, at the place where it's supposed to be hydrolyzed between the beta and gamma phosphate. Affinity was about nanomolar range. So we rescued, I showed it, that it rescued the activity of the oncogenic RAS mutants. And it meant mechanistically that the rate-limiting step of the RAS GTPase reaction is indeed involving the catalytic glutamine. And what RAS has to do, or actually GAP, when it binds RAS, is helping RAS to bring the catalytic glutamine to the right place. Because in RAS, it is very much away. When you look only at RAS without the GAP, it's much further away. And positioning of the catalytic glutamine is a major obstacle in order to get the GTPase to behave as an enzyme. Okay, so this paper was published something like 5 years after our study was published in PNAS. It's by the group of Arya Varshal, a Nobel laureate. And he also studied the RAS GTPase reaction, but this was in silico, of course, not in the test tube, not in vitro. And he actually got a very different conclusion, arrived to a very different conclusion that we did. What he did here is created mutations in the computer. So for example, this mutation is the famous mutation of the glutamine Q61L of RAS, and is looking at the energy of the different states along the pathway of the reaction. And you should look at these peaks here, which are the transition states. So if the transition state is going up in energy, it means that the reaction is slower, of course. You inhibit the reaction. And indeed, that is what is happening with the glutamine 61 when you mutate it to leucine in silico. However, he did something that can only be done in silico, and he made like a glutamine to glutamine mutation, which is non-polar, meaning that the nitrogen and the oxygen are now not able, it's like he put carbons there instead of nitrogen and oxygen. So it cannot form the polar interactions. And with this specific artificial mutation, it didn't get any difference in the energy of the transition state from the wild type. Both are here, not any significant one. So he concluded that the role of the catalytic glutamine is indirect. It just helps to organize the conformation of the active site, like the G12 is doing in RAS, for example. I don't agree with this, but I cannot say anything about the methodology. I'm not a computational biology. Our experiments, although I admit, are also indirect because we use a different substrate, but I think that they tell us more convincing evidence about what is happening in the reaction. What I could argue, for example, is what the supporting evidence that they showed in the discussion. They said that another source of support that the glutamine 61 acts in an indirect way come for the studies of the glutamine mutant of GS alpha that we studied, as I showed you. And in this case, he said that there is only a 10 to the second fold reduction of the rate by the mutation, while in RAS, it's 10 to the 6th effect by the same mutation of the catalytic glutamine. And if the catalytic glutamine was involved in a special direct interaction with a transition state, you would expect a similar contribution. Now, I completely disagree with that for several reasons. First, we observed a more significant, much more significant loss of activity. So you can see it again here. I showed you, where is it? Here. From 50,000 to less than 90, actually, I think that it's not 3 orders of magnitude that I show you here. It's probably 4 or 5, going all the way down to the minimal value that we can get. Second, the rate limiting step of the 2 reactions is simply different. So this conclusion cannot be made, because with Russ, I showed you, and there are other evidence also from the work of Fred Wittinghofer, more direct evidence, that the rate limiting step is the chemical reaction itself, getting the glutamine to the right place, and then the chemical reaction. While with the heteroterameric G protein, the product release, the phosphate release, is the rate limiting step of the reaction. So if you mutate a residue, and the effect, let's say, is 10 to the 6, but 10 to the 3-fold is just the difference between the 2 transition states, then you see an effect of 10 to the 3 and not 10 to the 6. I hope that I was clear about it. If not, I will explain at the end. So I would like to conclude this part with acknowledging, of course, first of all, Tzvi, the group of Alfred Wittinghofer, in particular Reza, who is originally an Iranian. It only helped us to have immediate chemistry between us. It was a beautiful project, and we stayed good friends. So we never collaborate after that, because I went to another direction. But we are still good friends. This is just 2 years ago when I visited him the last time. OK, so now I would like to go to, I think I will skip that, actually, to the work that I did in the postdoc. It related to the structure of Krebs-CBP interaction. And I will just skip it, because I don't think I have enough time. If I do, I'll go back to it. And now I will show you some of the work that we're doing in my lab. As I told you, we're working on macrophages, signal transduction in macrophages. Not everything is cyclic cAMP, but some of it is indeed cyclic cAMP. So basically, we're interested mainly in expression of TNF-alpha and IL-10. TNF-alpha is a key pro-inflammatory cytokine. It is immediately induced after the cells recognize any pathogen-associated molecular pattern, like LPS, for example, of gram-negative bacteria. And it is produced in great quantities, and it is responsible to a very large extent to the inflammation. IL-10 is an anti-inflammatory, key anti-inflammatory cytokine. So when macrophages are activated by LPS at the beginning, and this is like the send clock that shows you that it's the early phase, you get a lot of TNF-alpha, but very little IL-10, because you want inflammation now. Later on, you want to resolve the inflammation. So you get, at the late stage, you get an increase of IL-10 following the LPS, late LPS signaling, and then TNF-alpha goes down. So much more IL-10, much less TNF-alpha. Now, how is cyclic cAMP related to that? Cyclic cAMP is a key anti-inflammatory mediator in macrophages and in other cells, particularly in macrophages. So whatever you use in order to activate the cyclic cAMP pathway, and there are many endogenous ligands, of course, that do that through different receptors, you get a large increase of IL-10 already at the early phase of the reaction. Okay? You don't have to wait a long time. So actually, you dampen inflammation before it has a chance to start. And we're studying the role of the mechanism of how this is happening. And of course, we first hypothesized that it is going through CREB, which is the cyclic cAMP response element binding protein. And indeed, you can see here that if we silence CREB, the effect of isoproterenol on LPS-induced IL-10, which is it's going down from something like 9-fold to almost the red line, which is LPS alone. So we completely abolish the effect of isoproterenol on the on expression of the anti-inflammatory IL-10. And also, if you look here on the right, the pro-inflammatory TNF-alpha, this is induction by LPS in green, and then LPS plus isoproterenol, there is a negative effect of isoproterenol, of cyclic cAMP on TNF-alpha production. But when we use a dominant negative CREB, this effect is partially gone. Okay? It goes back up, and we did the same with siRNA and also with a stable cell line without CREB activity, or actually with reduced CREB activity, and we got the same results. What is the problem? Well, what was the problem actually? Many reports from I would say dozens of labs reported that LPS is mediating its effect partially also via CREB. LPS, according to many reports, was activating CREB. Now, if it was activating CREB, how can we further activate it with cyclic cAMP and see the effect of cyclic cAMP? That was one problem. The other problem was that we don't see this effect of, mediation by CREB for LPS, because when we use a dominant negative CREB, you see there is no effect on LPS signaling, just on the signaling of isoproterenol. And the same was here. It didn't go below the red line, which is the LPS activity. So we went on to test it, and I realized that in all of these papers, practically all of these papers, when they say that LPS activates CREB, they don't really look at activation. They looked at phosphorylation. And you can see also here in our studies, we also saw that LPS is causing phosphorylation of CREB at 30-60 minutes, and isoproterenol is causing about the same extent of phosphorylation of CREB. It's earlier kinetics, but that wasn't the issue. We discovered it later. So so how is phosphorylation related to activation? Everybody assumed that when you phosphorylate CREB, you get it active. But it's not so, and you can see it in the next figure. So this is a reporter for CRE, cyclic cAMP response element. And you can see that we get a beautiful activity with isoproterenol, which is blocked by the dominant negative CREB, but LPS does not activate at all this receptor, this reporter. So clearly, phosphorylation does not equal activation. So we published it with these three authors that were in this order, and luckily I found a picture that they are standing next to each other in the same order exactly, first, second, third author. I was the only one out of line. I was supposed to be fourth. But that was by coincidence. So I will summarize all the other experiments that we have done in this project. We found out that the difference between phosphorylation and activation results from the identity of the kinase, the CREB kinase. So for the cyclic cAMP pathway, of course, it's pKa. So you get phosphorylation on serine 133 and activation. However, with LPS, you get phosphorylation of CREB on the same residue, but by a different kinase, by MSK1 downstream to P38 and ERK, and that does not result in activation. How come? We wanted to study that, but actually someone was faster than us, the group of Arthur from Dundee. And they published this very elegant set of experiments where they showed that in resting cells, you need CREB to be phosphorylated in the nucleus and phosphorylated, but you also need this protein, which is the coactivator of CREB, CRT3. That's in its name, include the word coactivator. But it is constitutively phosphorylated by Cyc, which makes it cytosolic. The phosphorylated form is only cytosolic, so it cannot participate in this transcription. When you induce the cyclic cAMP pathway, pKa is activated and you phosphorylated CREB, but you don't change the situation of the coactivator. And that's why you don't get the transcription. Sorry, I made a mistake. Of course, when you do that with LPS, I will show it to you later, that is the situation. When you use a cyclic cAMP inducer, you activate pKa, you get CREB phosphorylated, and also you get Cyc phosphorylated. Phosphorylated Cyc is inactive, and therefore CRT3 cannot be further phosphorylated. Of course, there is a phosphatase that removes the phosphate, and then CRT3 can go to the nucleus and activate CREB-dependent transcription. Of course, when you stimulate the cells with LPS, you stimulate the activity of another kinase, MSK1. It's doing the same job on CREB, but it doesn't do anything to CRT3, and that's why we don't get CREB-dependent transcription. This was also, similar results to ours were also shown by the group of Mark Montmini at the Salk Institute, and he showed that pKa is causing the phosphorylation of CREB and the activation. This is an antibody for the CREB-CBP complex, so it means activation, the red dots here. While pKc, when you stimulate pKc with TPA, you get phosphorylation on the same residue, of course, but there is no activation, as we have shown in macrophages with MSK1 versus pKa. A second project that I would like to show you is the mechanism. Well, I will show you very little of it because we don't have time. So I will just want to tell you that the mechanism, we worked on the mechanism of IL-10 expression by the cyclic cAMP pathway. And you can see that both on the level of secretion of endogenous IL-10 and the reporter of IL-10 promoter, so transcription. This is even more impressive. You can see on the transcription level there is a huge synergism between cyclic cAMP and LPS at the early phase. Okay, I don't show you here ISO alone, but ISO alone is very, very low, and here it's completely zero. But LPS has a very small activity, as expected, at the early phase, because you don't want anti-inflammatory process to go on at the early phase. However, when you co-stimulate the cells with cyclic cAMP, you get huge synergism, and then it goes down until at 24 hours, there is no effect at all for cyclic cAMP. So we've done a lot of experiments on this. It's summarized in 2 papers, actually. One is under revision. The other one will be submitted as soon as the first one is accepted. And there is the model that we now propose is that we identified 2 CRES sites which work in tandem. They are in tandem, and they work cooperatively on the promoter of IL-10. And they also work cooperatively with an SP1 sequence that is working constitutively, actually. There is also an LPS effect, which is very, very low, but it is crucial for the synergism with the cyclic cAMP signal. And that's why you get a lot of IL-10 in the early phase. At the late phase, LPS is inducing an autocrine loop of secretion of type 1 interferons, interferon alpha and beta, which are acting on the same cells or neighbor cells in order to elevate IL-10 induction by overstimulating or doing an extra job on the constitutive activity of SP1 is amplified very much, and now it's sufficient for full activation of the IL-10 promoter. And now if we mutate the CRES sites, we get no effect. Okay, we add isoproterenol, we don't get any effect. So these are the people from my lab who worked on this project with the help of Ian Fraser from NIH also in one of the experiments, and Doron Gerber in another experiment.